In 2018, a Chinese scientist did it. Edited the DNA of human embryos. They were born. He went to prison. And the technology that made it possible has only got faster every year since. Are we ready for what's next? Today, we put human gene editing on trial, the case for, the case against, what actually changed in the last decade, and the verdict that lets us steer this instead of being run over by it. First, the case for, because the moral case for editing some genes is genuinely overwhelming. Around 6,000 genetic diseases exist. Many are devastating. Many are pediatric, cystic fibrosis, sickle cell, Huntington's, Tay-Sachs. Edit the relevant gene before symptoms emerge. And you could prevent a lifetime of suffering permanently. And it's already working. In 2023, the FDA approved Casgevy, a CRISPR therapy for sickle cell. Patients who lived in chronic pain for decades now don't. This isn't theory, it's a treatment, the first of many. Beyond rare diseases, gene editing could reduce risk of cancer, Alzheimer's, heart disease, not eliminate, reduce. Across a population that's millions of lives extended, tens of millions of years of healthy life without illness. And germline editing, editing reproductive cells, would compound across generations. A single edit made once persists in every descendant. The good versions of this would functionally end certain diseases within 100 years. But the case against isn't just caution. It's a serious reckoning with what happens when an irreversible technology meets the world we actually live in. Today, the cure for sickle cell costs over $2 million per patient. Genome enhancement, if it ever existed, would be even more expensive. Edit the children of the rich first. Their kids start with biological advantages. The inequality doesn't compound, it locks in. And it doesn't stop at disease. The same technology that prevents Huntington's could one day select for height, intelligence, eye color, athletic potential. The slope is real. Every regulatory line that was supposed to hold has been crossed. CRISPR is precise, not perfect. It edits the target gene, sometimes also nearby ones. In an adult, you can monitor for cancer. In an embryo, the off-target effects propagate into every cell, every tissue, every descendant, forever. Drugs can be taken off the market. Bridges can be torn down. Germline edits cannot be undone. Whatever we get wrong, and history says we will get something wrong, it stays in the human gene pool. Indefinitely. In 2018, Jiankui edited 2 human embryos to be HIV resistant. They were born as Lulu and Nana. He went to prison. The babies are reportedly fine, but the line was crossed. There is no putting it back. So is gene editing inevitable? To answer that, you have to know how fast the tools are actually moving, and it's faster than you think. Sequencing a human genome cost $100 million in 2001. Today, it costs $200. CRISPR followed the same curve. Editing a gene now costs less than a tank of petrol. The tools moved from national labs into university basements, into garages. Meanwhile, regulation is 15 years behind. Different countries have different rules. The US allows somatic edits, bans germline. China is more restrictive on paper. Some countries have no laws at all. The technology routes around the strict ones. Which brings us to the verdict. And it isn't ban it or allow it, it's something more useful. Editing the genes of a sick adult is medicine. Editing the genes of an unborn embryo for traits is something else entirely. The first should be allowed and accelerated. The second needs the slowest, most international, most careful framework humanity has ever attempted. The right question isn't, should we edit DNA? It's, who decides, in what cases, with what reversibility? Because the answer to that question is what makes one path medicine and the other path eugenics. 3 rules. Edits to existing patients with consent. Yes, edits to embryos for disease prevention. Only with international consensus and decades of safety data. Edits for enhancement, not until we've genuinely solved inequality, which is to say, not soon. Gene editing will define the next century the way nuclear weapons define the last. The tools are not the question. The choice is whether we build the international scaffolding now, or repeat every mistake of every technology we ever invented in a panic. What's your line? Where would you draw it? Drop a comment. The hardest thing about this debate is how few people have actually thought it through. If this helped, hit subscribe.